Hello, my name is Laurent Servais. I'm professor of Pediatric Neuromuscular Disease at the University of Oxford, and I'm going to discuss with you the trial that is coming in the coming weeks or month in the UK, which is called KICAS, the phase 1-2 clinical trial of GTX-102-001. So, first thing first, and a disclosure, we're going to talk about the trial, not about the treatment, not about the cure, about the trial. And a trial is a trial, which means that the aim is not to treat a patient, it's not to cure patients, it's to answer a question. Does it work? And is it safe? And that's it. And in a phase 1-2 trial, actually the main question is it safe? So I think that it's important to consider when we start speaking about a trial to appreciate that if you take part in a trial, it's not only for your child, it's because you believe that things must move forward for the entire community. So the drug which is investigated here belongs to a large family which are called the oligonucleotide antisense. And these are new drugs, but not that new actually, because the first drugs were approved actually more than 22 years ago, right? And there are several drugs that have been approved over the last years. And myself, I have a quite large experience with some of these drugs, especially Spinraza, which, as the GTX-101, is a drug that is intrathecally delivered. So it's the same mechanism, the same type of drug, the same family of drug, but also the same route of administration. So this is how it looks. This is actually something that looks like RNA, but which is not RNA. It is much more robust than an RNA, and it allows to interfere with the splicing process of an RNA. And I won't discuss again the mechanism of action because there are, as explained it in a lecture. And so GTX-102 has been investigated in monkeys, in non-human primates, and a single dose demonstrates a robust knockdown of UBF3A AS in key brain regions, which leads to paternal UBF3A RNA expression. And the interest here is to see that actually the drug goes in the entire brain. So we're going to discuss about a phase 1-2 study, which means a study of which the primary objective is to demonstrate safety. And the study has already started in the UK, in the US. It was a first in human study with 4 monthly doses in a dose escalations with several groups and several different doses. And as I told you, GTX-102 was delivered as with a lumbar puncture, and there was no placebo actually. So the primary objective of this first study that happened in the US was safety, right? And then there were secondary objectives like pharmacokinetics, which means how much after injection do we find drug in the CSF and in the blood. And there was an exploratory objective, which was efficacy. So first safety and then efficacy. So the key inclusion criteria of this first study in the US was deletions and age between 4 and 17 years at screening, stable seizure control, normal kidney and liver test, and able to tolerate anesthesia. Actually, it doesn't mean that the drug cannot work for uniparental disomy or for point mutations, it doesn't mean that the drug cannot work for children younger than 4 years or for young adults. It's just that when you conduct a trial, you first need to go on a very well-defined populations and try to get population that is homogeneous. And when it comes to a trial, you have inclusion criteria and you have exclusion criteria, and you cannot play around. I mean, if it's 4 to 17 years, it's 4 to 17 years. If the child is 1 day older than the inclusion criteria, you just cannot include these patients. So there were several exploratory endpoints for the efficacy, even if efficacy was not the primary endpoint. And you know it better than me as parents, that when it comes to Angelman, we need to investigate all different aspects of the disease, like sleep, seizures, communication, behavioral, cognition, motor function. And all of this was actually investigated using a battery of outcomes. So this is what happened in the US. Actually, there was 3 cohorts. In each cohort, the dose, the starting dose was different. And as you can see, in the first cohort it was 3.3, but then they rapidly escalated to 20 milligrams. And in the second and the third cohort, the dose was much higher. And then happened adverse events. In all patients, there was a transient ataxia and fatigue, which was dose-related. It was primarily observed in patients who received 20 milligrams and more. It was of a variable severity from one to another patient, and it started very quickly after the injection and lasted for 1 or 3 days, right? And then patients could go back to the normal previous way of functioning, and anesthesia was quite well tolerated. This is what we call an adverse event, and it's not serious because it doesn't lead to hospitalization, it doesn't lead to death. It's an adverse event, but there were also serious adverse events. And in all 5 patients was observed a mild to moderate lower extremity weakness, and it occurred later, 6 to 30 days after the last infusion, with significant weakness in 3 of the patients and inability to walk in 2 patients. Of course, when it happened in a trial, you need to stop, right? You need to be sure that to understand what happens. And the good thing is that this was reversible, and patients retrieved their normal functioning. But nevertheless, 2 of the patients during several days were unable to walk, and this was really related to the highest dose. So actually, it was related more than probably to inflammation of the nerve around the injections, which you do not observe with other antisense nucleotides, and was really related to the dose. And at the same time, we could observe improvement in all patients, and this is the clinical global impression after 4 months of treatment. And as you can see, there were quite a significant improvement for fine motor, communication, behavior, and that is the gross motor function that was actually declining in these patients. It means that maybe we have some efficacy, and I say maybe because we're talking about 5 patients without placebo, but it's sure also that at the highest dose there is a real risk of safety issues. So the UK protocol that is starting with the approval of MHRA, and we still need to get some the final approval of the ethics, will start with much lower dose, 3.3 and 5 milligrams. and there are 4 monthly doses over 3 months, and then maintenance over once every 3 months. And actually the final dose will be also dependent upon the absence of safety issues and also signal of efficacy. As you can see, this is quite a significant protocol in terms of burden, right? They are the injections, but there are also several tests at very precise day, and this is something important to consider when you take part of a trial. I mean, if the next injection is, for instance, at day 44, day 30, I mean, this is day 30, right? And it's not possible to say, well, maybe next week or the week after, I cannot do it this week. No, if we are in a protocol, we must carefully follow the schedule of event. So that's all the tests that are planned in this protocol. And as you can see, in the maintenance dose, patients will receive the drug every 84 days, so a little bit less than every 3 months. How is it going to happen in terms of anesthesia? We cannot do it without anesthesia. This is just impossible, and we need to have either an anesthesia, either a sedation. We will try to do our best to make sedation only, and there is a paper that has been published in the US by a team that is also performing this trial that shows that actually in another disease, which is Niemann-Pick, in which we face the same issue, we could afford with inhalation, induction, and mask maintenance in patients, and only limited needed to get an anesthesia. So these are the place where you can find the information. If you wish to connect with me as the coordinating investigator in the UK, I will be more than happy to give you additional information, and these are the clinical sites in which you can find more information. Then we have received several questions before the symposium. Is there a specific age group? When will all the patients be included? First, we need to know what are the data in this first trial before considering other patients, but obviously there is no reason that they could not be at one point some extension, but the very first step is to gather data in this well-defined population. Which genotype? When will other genotypes be included? Later. And we must consider that the better we know the natural history of Angelman, the better are the data that we have in our hands for natural history, the easier it will be to bridge the data that are acquired in a specific age group for specific mutations and the larger group of patients in terms of age and in terms of mutations. Is there other trials coming? Yes, there are many others, and we need to be ready. We need to improve our clinical trial readiness. In the UK, can I put 2 of my children forward? Sure, you can. Just connect with us. Will there be a cost? No. A trial cannot cost you anything, and there is also reimbursement of any fees and of your time. When will this trial be available in the UK? We are working hard to make it happen in the autumn. How many children will be involved in the UK trials? Well, it's up to 12 maybe, because it's a trial that will take place in the UK and in the Canada, and probably Canada will start a little bit before the UK. If successful, what is the time frame for the drug to be available? This is just impossible to say, and probably it is at the bare minimum between about 4 years before it becomes NICE approved, but this is a pure speculation. Will participants be able to receive the drug after the clinical trials? That's the norm. It means that if a drug is efficient and is working, and that we feel that the clinical development must continue or that approval can be obtained, the rule is generally that patients keep on medications. Of course, if it doesn't work, it is better for everybody to stop. Can this type of treatments help adults? Probably, but one more time, we will need to bridge the data we get in children into adults. And this is an additional reason for which we need the natural history study. What will convince NICE to recommend NHS to offer this drug? The quality of the data, the quality of the study, and the understanding of the epidemiology in the UK. I can tell you that is primarily important. What will happen with potential improvement as the drug wears off, like 5 new words have been learned? Will they be forgotten? We don't know. We just don't know, and we need to see. How many times per year will a top-up be needed? Actually, it's every 81 days, so nearly 84 days, so nearly 3 months. What if there is a more potent or efficient drug available? Actually, the investigator must inform you about other options. Sometimes it's not possible to go in a trial if you are already on another medication. It's very dependent from one to another treatment. There is already a global Angelman registry. Does it cover the same question as the upcoming NHS would? Certainly not. Both are important, but they don't cover the same kind of information. A natural history is something prospective with well-defined outcome by the same team, and the global Angelman registry is very important to have a kind of broad view. So we need both, and we need the natural history certainly to pave the way to more clinical trials, to get faster the NICE approval, and also to be able to bridge the data that we have in a limited populations to other phenotypes, to other genotypes, and to other patients. You have my address in the slides. You have the address of Dr. Marchetti. Please feel free to connect with us. And the very last thing I want to say is that I truly hope one day we'll have one or several drugs for Angelman. And today we have several drugs for SMA, and the time for newborn screening has come for spinal muscular atrophy. Unfortunately, it's a slow and painful process in UK. We have initiated a parliamentary petition. Please sign it. And I really look forward in 10, 15 years maybe to do the same for Angelman. But let's show our government that we care about rare disease. So please, if you're convinced, go on the site, sign it, and forward the petition to your contacts, to your friends, promote it on social network. Thank you for your attention. Please feel free to touch if you have any questions. Thank you.